we presented data on a mouse model where we tried to study the impact of chronic leukemia, of CLL, on an antigen-specific immune response. And the reason of doing so is that there is a lot of evidence now showing that in the context of CLL, T cells are dysfunctional and are therefore poorly activated as part of cellular immune therapy, like CAR T cells or bispecific antibodies. And the problem is that we don't have a clear idea why this happens, what CLL actually imposes on those T cells. And it's a very difficult subject to study in vitro because it's you don't know exactly what kind of what the antigen specificity of those T cells, and you need actually a dynamic model to do that. So what we did is we developed we adapted the mouse model, the TCell1 mouse model, which is a mouse model very much resembling, specifically if you look to the immune system of CLL, where those mice have a very late onset of a B-cell lymphoproliferative disease, and also those mice develop T-cell dysfunctions. And what we then do is we use an adoptive transfer model, so we inject the splenocytes of aged mice, bring them into new mouse, and then it takes 4 to 6 weeks to get also a CLL-like disease. And at the moment they got that disease, we gave the mice other T cells, so-called OT1 cells. And OT1 cells are very restricted T cells that only recognize ovalbumin as antigen. And what you can do then is you can infect the mouse with an ovalbumin expressing virus, and we took MCMV for that. And by doing so, you can actually follow an antigen-specific immune response in yes or no the context of CLL. So the control mice were just normal mice that we also gave these OT1 cells, and then the MCMV ovalbumin. And the study was for mice that had already developed CLL. And what we saw was quite remarkable, is that although the number of T cells and the number of effector cells after 7 days of infection was completely equal, if you take a more deep dive into those T cells, you see that in the context of CLL, you get much more inflation of so-called short-lived effector cells, which are cells that quickly go into apoptosis, while in the wild-type mouse, you get an inflation of MPEGs, memory precursor effector cells, which are the cells that you actually want to have if you want to have long-term remission, for instance, for CAR T-cell therapy. Also, when you look to the function of those T cells, they were different. We saw that in the context of CLL, cytokine production was lower, specifically interferon gamma, and also that degranulation, a marker of cytolytic activity, was also lower. That data led us to the question, so how is it regulated? And we know for T-cell abnormalities that much of this is regulated at the epigenetic level. The impact of CLL cells could be very much epigenetically regulated. So we did ATAC-seq, which is a technique to get the epigenomic landscape. And very shortly said, what we saw is indeed that the presence of CLL suppresses key transcription factors which are relevant for memory formation and activate cells that skew the T cells into an effector state. And that's a bit where we are now at this study, but I think it gives a lot of new insights in how CLL cells actually impact T cells, and might also hold clues how to prevent this, to also make this kind of cellular immune therapy available for patients with CLL.